Good morning, everyone. In yesterday's class, we were discussing about several protocols about screening. Screening is about how to find out the gene of interest from a given lot of clones, for example. So in that, we have discussed about colony hybridization. It's almost like southern blot, except that we are using a culture plate instead of electrophoresis gel. And then we make a blot, then we do hybridization, and then we can figure out which one has the gene of interest. Similarly, if in a gene library, many a times they are used, phage vectors are used. Phages form plaques, and to how to identify them is almost a similar technique, and it is called as plaque lift. And then we will identify whichever the plaque is having the gene of interest, and then we will clone it as per requirement and proceed. I mean, we'll identify and then proceed with what to do with that. We also discussed about chromosome walking. Chromosome walking is about identifying the order of genes that are present on a chromosome. When we don't know the sequence of the the order of the genes or the sequences, how can we find it out? And for that, there is a technique called as chromosome walking, in which if we had some sequence information, we can use that and identify a clone, use that as a probe to identify clones sharing or having overlapping sequences, and so on. You can find out the sequence, the clones, and then we can always characterize each of them, and then be able to figure out the whole sequence of the chromosome, for example. And that's called as chromosome walking. Then we also discussed about subtractive hybridization. Subtractive hybridization is about subtracting the transcriptome of one cell from that of another sample, from one sample to the other sample. It is about finding out the genes that are expressed in the test and those that are not expressed in the control. So as I told you yesterday, I hope you will remember that it's not only about treatment. It can be used to do several things, like it has one thing is, first thing is treated and untreated cell. Untreated and treated is one way you can do. You can collect the mRNA, make the cDNA, and then identify. We can also do something like cell type A and cell type B, right? These are from the same organism, from different lineages, and in that case, we can identify as well. So similarly, we can compare the transcriptomes of any two cells. It's not about comparison. It's about cloning, being able to clone the genes that are differently expressed genes. You can say it like that. Okay, that's how that is what it means. So I hope you remember that we have discussed a lot of things in that: driver mRNA and tester. Driver is something like the control, and tester is the one that we are identifying. So you can think of it like that: tester mRNA or cDNA minus driver mRNA is what we are going to clone. We are cloning the genes that are different from, that are present in tester but not present or not expressed in the driver, something like that. So in that we will make cDNA, but for the driver we will use biotinylated primers, so that we'll get the driver mRNA, that is cDNA, that has biotin on either sides, like this driver, and cDNA for the tester. We'll just get the cDNA. Now we'll mix them, denature and reanneal, and we will also take 10x or higher concentration of the driver. So we will increase the population of these things. Whichever are common genes, they will either form um, driver or tester and driver, if because we are taking higher in concentration. The unique ones, those that do not have a counterpart in the driver, will reanneal, and they are tester cDNA. And then using streptavidin, we can eliminate all the genes, all the cDNA that have this biotin, and those that are unique can be now cloned. And we can directly get the sequences that are different, that are present or expressed in tester, but not in the driver. That is about subtractive hybridization. Okay, now we are going to go into differential screening. It is to identify the genes expressed. It's not about cloning necessarily. We'll take libraries, we'll see it in the next slide. It is about screening, right? To identify the genes that are expressed in here in cell type B, for example, but not expressed in cell type A. Okay. Subtractive cloning is we are cloning those genes. Here it is, if you want to make a large scale comparison between two different cell states or cell types, then you can use this protocol. So I hope if we learned about the colony hybridization, and if we understood the subtractive hybridization, the logics, it's almost like a fusion of these two. So here is a library of genes. You can think it of as gene library. And now we want to know, we are making gene library has all the genes, right? All the sequences expressed in a in an organism, right? It is a representative of it's a set of clones that are representing all the sequences in the genome. That is a gene library. Now we can do something else. We can differential screening of a gene library. So what we do is we take sample A RNA and sample B RNA, and then we are making cDNA from both of these things. So if I will just go back a little bit to have a background for this experiment. If there is an individual, as I already described before, genome is almost same in all the cells. Genome is same in all the cells. We have about 210 different cell types in our body. Although we have 210 cell types, all the cells the genome is same. There are exceptions, but majority you can think of it that in all these cells, the genome as a set of the total set of genes is same. What is different among them is transcriptome and proteome. Transcriptome and proteome will determine how the shell should look like, the morphology, the form, and how it should function. So genome is essential in the sense that the gene should be present in the first place. The transcriptome and proteome will determine when is it expressed and how much it is expressed. And the total set of proteins and genes expressed, the total proteins and RNA will determine how the cell should look like and how the cell should respond to the environment or to the signals. Okay, form and function is determined by these. So in the protocol that we are going to see is they are making the objective. Let's discuss the objective based on the previous slide, the slide that we are going to see. The objective is this, or the logic you can think of it. The whole genome is like this, and there are genes A, B, C, D, like that. I'm just making up for the sake of illustration. And I have cell type A, and I have cell type B. Sure, because their form and function are different. That's why we are calling it as cell type A and B. That means their transcriptome and proteome are different. And I want to know which genes are common in both of them. I want to know which genes are present in B but not in A, and which genes are present in A but not in B. So if we know these, the differences between the in the transcriptome and the proteome, I mean, if we know that these cells are different, that means they must be. It is because of their proteomic and transcriptomic differences. And if I find out what these genes are, then I can have a good idea about what genes and how the genes are related to the function of the cell. So the cells expressed in B but not in A are likely the genes expressed in cell type B but not in A are the genes specific. You can call them as B specific, we'll say B cell type specific genes. Okay, I'll write it that way. These cells will be B cell type specific. That means they must be producing some gene proteins or enzymes that are essential for the form and function of cell type B. These genes here must be A cell type specific genes. The genes that are expressed in both of them could be housekeeping genes like metabolism, glycolysis, and so on, right? So in this protocol, that is what we are trying to see. Let me finish this. Say if we had some things like this, if we had in both A and B, you may have A, C, and D are expressed in both. That means they are involved, probably they are in the genes. Genes A, C, D are expressed in both cell types, then they might be involved in those kinds of functions. And if we had B expressed only in B in this one, that means the gene B is important, it's specific for cell type B. And if we had a different one, uh, like this, E and F are expressed here, that means those genes are A cell type specific. So let's now try to find out. So we take the gene library, and then we have plated them here, and we are making duplicate filters. That means we take a filter, make a print of it on one filter, take another one, and make another filter. So we have two filters here. Okay. What we are doing with the sample is we will take sample A, RNA from sample A, and RNA from sample B, and then we are making cDNA library with the cDNA. We made cDNA that are labeled. It is radio labeled, and I hope you remember. Should we use gamma labeled ATP, gamma phosphate labeled ATP, or alpha labeled alpha phosphate labeled ATP? Which one do you think we should use? Anyway, DATP. Which one are we labeling? Any idea? Should we use gamma ATP or alpha ATP? That is the point of the question right now. I would want you to look at how to make probes. That is essential because I might ask in the CA exams. Okay. We will also have a few questions, practice questions in one of the classes before the CIA. So you'll get an idea about what kind of questions to expect, and then you probably will be able to solve it better. Okay, the one that we will use is alpha labeled, because during polymerization, alpha phosphate gets incorporated, whereas beta and gamma will be released out as pyrophosphate. So we would, if we have to use, we will use, if it is random labeling, we would have used phosphate labeled nucleotides. Okay, so we have labeled the mRNA, the cDNA from sample A and sample B. And now in one of the filters, we would add the cDNA label and try to do something similar to that of colony hybridization. So cDNA, if the gene is present, then the cDNA will bind to that particular sample, to that clone, right? And the same thing we will also do with sample B here. We will hybridize with one of the other filters. Now the comparison starts. Say this gene that I'm pointing at, this one and this one are expressed in both, right? That means it falls in the category, probably it's housekeeping or some essential component of the, for every cell. The same way you can see, identify majority of them are actually common in both of those, right? And you might find some signals like this one, the blue ones that are that are giving hits, which are not present in sample A. That is, that means those are the sequences or genes expressed in B but not in A. And then we can always go to the master plate, pick those colonies, and identify what they are. And that gives a good idea about the genes that are expressed in, we can identify the clones that are expressed in B but not in A. That is the differential screening of gene library. Okay. Any questions here? Okay, let's move on. There are, as I discussed, when it is screening, we have used hybridization. Hybridization is based on homology of target and probe, right? So our everything is about probe, and this is nucleic acids binding or base pairing, and that is how we signal is identified. What if we didn't have any sequence to make a probe? What do we do then? We only know that there is a protein, and we know that the protein is produced and it has a certain function, and we are interested in a clone that is producing the same protein. We can use almost this similar technique. We can also use differential display that we have discussed already on the phage surface display, right? So you also have to remember that is also one of the screening. You can use it as a screening method as well. So in this case, what they do is they take a polyvinyl sheet to which IgG is IgG antibody against that particular antigen. Here, the protein of interest is the antigen. So we should have had, because we don't have probes, but we don't know the sequence. That's why we don't have the probe. So we can identify it using antibodies. And for those antibodies, we can take it into test animals, usually rabbits or horses, and they inject the protein into the bloodstream. And after some days, or with booster doses, the animal would raise antibodies against the protein. Anti-P, I will call it as. Okay. So anti-P antibodies are raised, and those the IgG type. Okay, IgM is the first one, and IgG is the most effective one. So we would have had antibodies which will bind to that like this. And so one more thing you need to remember is we have antigen, and the antigen will have multiple epitopes. Let me rewrite it. What is epitope? Epitope is the region to which the antibody binds, right? Here, sorry. Here I have an antigen, and each of these regions that I'm putting it here, this is one epitope, this is another epitope, and this is another epitope. So antigen can have multiple epitopes, in the sense that, in the sense, in the body, when this whole antigen is injected, antibodies can be raised against this, and they will bind to, and the antibodies will bind to it, something like this. So we need to have antibodies. Monoclonal antibodies are antibodies against one epitope.
You went to the specific epitope, the two.
Yes, that's right. Monoclonal antibodies are, they are all specific to the same epitope. They are coming from one clone. So one clone would have one type of antibody, because if it is one antibody, then they all are binding to the same epitope. So here we have a polyvinyl sheet to which IgG antibodies, the antibodies that I said, these will be attached. Assume that they are they're binding to X epitope. This is Y, and this one is Z. Assume that they are binding to the epitope X. Okay. So when you allow the protein or the cell surface phase display, or so they allow the protein to the apply the sheet on top of it, and some of the proteins, if they are bound, they can be bound to the antibody here. And now we have many antibodies as well, right? In some places they are bound, in some places they are not bound, wherever it is not present. Then we have a second antibody for a second epitope, something like Z, for example. Sorry. Then when we add that antibody is labeled with iodine. Okay. The antibody present here is not labeled. It is just for binding to the antigen. And then we have labeled antibody, second one. You can think of it as IgG, as anti Z, okay, to this epitope. The question I will have is, or why I actually have this slide is that, what if we just label this one? Why not label just this one? Why do you want this antibody? Sorry, anybody? So if I see this polyvinyl sheet as surface like this, and I have added this antibody here, right? So antibody would have been present all over. I am giving you a reason, or trying you to visualize, what if I have had this antibody labeled? What is in this condition? So in that case, the antibody is already present, and in some cases, you may have the antigen is bound here and here. When you develop the, sorry, sorry, okay. When we have this antibody developed, again, wait a minute. Okay, sorry for the interruption. Yes. So I gave you a polyvinyl sheet which is having labeled antibodies, the red spots here, and I said that these are the location where antigen is bound. If I develop this sheet, what do you expect of the signals? Where all do you expect signals? Anybody, please? You'd expect signals everywhere, isn't it? Why would you expect signals everywhere? Because if you put labeled antibody like this already on the sheet, then the signal will be all over the body, irrespective of whether the antigen is bound or not. Now consider a situation different. You have a sheet where the antibody is present but they are not labeled, and now you have the antigen is you allowed the polyvinyl sheet to bind to the antigen, so antigen is bound here, here, and here, say for example like that. And then we added a second antibody, and that second antibody is labeled. Where would the second antibody bind? It will bind only to the places where the protein is present. And if we developed this the signal from here, what we will see is that we will only see the signal, we'll only see the point where we'll see the signal where only the protein is present. And that is the reason why we are using two antibodies and why the second antibody is actually the labeled one. And why not the first one? It's just trying to reason why this arrangement is made like this. Okay. That is about immunological screening. So there are, I will take two more. This is little bit of deviation from what we normally have. It is called RACE, rapid amplification of cDNA ends. Okay, rapid amplification of cDNA ends. That's about RACE. So it is something like this. There is a gene. I know the nucleotide sequence of a small bit, this part, but I don't know the rest of the sequence. How can I clone it? Right? For to clone the complete gene, how can I get primers? I cannot get primers. All I have information in my hand is that I know a small bit of this sequence. So how to do that is the question. We cannot directly use it as DNA because, or it becomes easier if you're trying to make a cDNA. That is a possibility. The objective I told you. If we have a long gene, for example, which we only know a partial or a small bit of sequence, which is not on the 5' side or 3' side, how do we clone it? So there are two ways. There is one way is the logic of the 3' and 5' race, they call it is. When we try to clone this region, then we call it as 5' race. We are amplifying the 5' ends. If we are making this region, then we call it as 3' race. If we have made these fragments, then it's not a difficulty to make the whole of it. Right? So these are the two things that we will discuss now, 5' race and 3' race. One of it is easy. 3' race is easy because if we know the mRNA, if we have the mRNA, we know at least the one side of sequence, right? It is poly A tail. We are already saying that we know a little bit of the sequence. Then all we need to do is make a primer of this with complementary this region and amplify it. That's easy. What about 5 prime race? That becomes difficult. We know one side, we don't know what is the sequence on the other side. So that is these are the difficulties, and the objective is this, and how do we solve it? 5 prime race and 3 prime race. 3 prime race is simple and easy. We can oligo T, and we can also add a, a linker if we want, like this, and we will generate the first cDNA strand. Like, if we remember cDNA libraries, that is the same thing. First cDNA strand can be made using oligo DT, reverse transcript, reverse transcriptase. And this region is the anchor primer. That means we can use that sequence as a primer for the next round of PCR. So it's not so big deal. And we got the first cDNA strand. Now denature, and denature using alkaline conditions, the RNA is lost. Then we are making another primer. The primer that I said we had information about, something like this region, right? So because we know that sequence, we can make a primer based on that and synthesize the second cDNA strand. So that it is straight and a simple protocol. So later what we will do is we will amplify using anchor primer. Okay. Anchor primer, say when you synthesize this first cDNA strand, all the mRNAs that have any mRNA or RNAs that have poly A tail will be amplified. Or not amplified, at least the first cDNA strand will be synthesized. We are only interested in one particular gene. It's not a library of, we are not interested in cDNA library. In that case, we want to, when we amplify the second time, in this round, we got the second cDNA strand of only the gene of interest. Why is that? Because the primer that we are making is specific to the gene of interest. So we only, the second cDNA strand we will only get for the gene of interest. We are only interested in that one, right? And next time, so when we have this cDNA strand, we will have, I will put it as XXX, which is this region, a new sequence. And now we are going to add antibodies against, sorry, the primer against XXX. And the cDNA strand will be synthesized. Second, the lower one, new one, anytime that is synthesized, will be only of the gene of interest. In this case, when we used oligo DT, we had all the genes, sorry, all the mRNA are first cDNA strand is synthesized. But from here on, only the gene of interest, that the 3' side will be expressed. So using PCR and internal primers, we can amplify it and then go for cloning. Sorry. Okay. That is called as 3' race. It is simple, straightforward, because we already have the information on one side and also the information on the other side, because as we established that we know a small sequence of the gene of interest. Then the sorry. Then the 5' race is something a little bit critical, and yes. So in that case, we have, what do we have to start with? We have the information of a small sequence. This is the whole gene. How can we clone this region is the question. So we take the mRNA and we use the gene specific primer, the one that is shown in the as a purple arrow, and we will extend it. So we will get first cDNA strand of gene of interest only, because we are using a a primer specific to the gene. It will only synthesize the first cDNA strand of that one. by using reverse transcriptase and so on. Then we have the first cDNA strand here, and we are using terminal transferase. Here they have used a DATP. So A will be added on both the sides, and here also. This is, of course, the RNA, mRNA. By using alkaline conditions, we can get rid of it. So what we are left with is the first cDNA strand, the 5' end of the gene of interest. Because we did homopolymer tailing, right, using DAA, we can now, because we know the sequence, so we can add poly T, oligo DT as the primer with the anchor region or for additional rounds in the future. And we can synthesize the second cDNA strand of the 5' mRNA of the gene of interest. So now we have cDNA, double-stranded cDNA of the 5' region. We can then use internal primers and the primer here and this other primer, anchor region primer, and extend this, and then we can clone it. Okay, so why when we do these things, how do we get the whole DNA? Sorry. So using, okay, let me draw the problem and then we will try to address it again. So this is the gene. All I have is information about this much region. I have the gene information. How can I clone the whole of it? Right? We used mRNA, and using the 3' race, sorry, 3' race, we used oligo DT and the internal primer based on the sequence, and we were able to generate a fragment like this. So I'll put it as double-stranded DNA. Then it will be easier for you to understand. So I put it as double-stranded, and this, the lower strand would be 5', and this is 3'. And then we got double-stranded DNA, this much to here. So sorry, using 5', 3' primers, we got this. This is 5', 3', 3', and 5'. This is using 3' race, rapid amplification of cDNA ends. Using 5' race, we would have gotten sequence some from here to here, right? And that would be 5', 3'. Now I will mix these two race products. This is at 5' race. I mix these two products. I'll mix them, denature them, and re-anneal them. So there we can have different types of products, out of which the hybrids would be something like this. You can see this top strand at least will be able to base pair with the bottom strand of the red one, isn't it? So we would get something like this. So sorry. Yes. Yes. Let me put the, now this is the one I'm writing here, that is 5' to 3'. And then I'm putting this one here, that will be 3' to 5'. The lower strand, the top strand here, that is the 5'. Is that right? 5 prime to 3 prime. And the bottom one here is 3 prime to 5 prime. Is that correct or wrong? Okay, that is a mistake, right? Is there a mistake? There is a mistake. Yes, now we are fine. The top strand is here, right? 5 prime, and this is 3 prime. The bottom strand, this one, is going to be here. The common regions, I'm trying to anneal only the common regions. This is where base pairing would have occurred. The same case is here. Now, how can I get double strand from this? Anybody? How can I get double strand DNA? S. If I add DNTPs or DNA polymerase and DNTPs and allow the extension reaction to happen, I can get this one, the 3 prime OH. From here, it will add, and then I will get a whole region like this. And this is 3 prime, and there is a template, so the whole region can be made this way. And then now I have whole cDNA of the gene that I'm interested in. For the product on this side, nothing will happen. Why is that? It's because extension only, polymerization will happen from 5 prime to 3 prime direction always. The 3 prime OH is important, onto which more and more bases are added. Here, and DNA polymerase would require a primer template junction. So this is primer, for example, this is the primer, and we have a template here. Then this should be the 3 prime OH, and then more and more bases would be added like this. But in this product, in the second product, this is 5 prime, and DNA polymerase cannot do anything about it. What we'll get here, this is what the product is of interest, which will then we can clone them and then study about it. Okay. Okay, in the next lesson onwards, we will discuss about how to analyze gene function, and that becomes important. And in CAA2, until gene function, whatever we left from CAA1 until whatever we are going to finish by the end of gene function, all these things will come as a syllabus for CAA2, and we will have some practice questions which will be of use to you all. Okay. Any questions here? Any doubts? And did we fix any date for the CAA? I'm still waiting for you people to say what would be the date.
Sir, as of now, no doubt, sir, and we are still a little bit more discussions are left, sir. Like November 2 is okay, but time we are asking for the timings alone. So if that is fixed, we'll immediately let you know, sir.
Okay. Yeah, good.
Yes.
Thank you very much.
Thank you, sir.
Yes. Sri Shakti Ashwin, can you call me, please?
Yes, sir.
Yes, please. Go. Bye. Thank you. Thank you, everyone.